This is biology test 2, and question 1 is saying, what role does a coenzyme play in an enzyme catalyzed reaction? The first thing that I want us to understand is the function and what really an enzyme is going to do. When you have got an enzyme, say this is the structure of an enzyme, okay, like this. This is an enzyme. An enzyme attaches to what we call a substrate. A substrate should have a shape or structure that is complementary to the enzyme. In other words, it should be able to fit into the enzyme. This part of the enzyme where the substrate fits, we call it the active site. And then the other parts of the enzyme, we call that as the allosteric site. So these two sites, you have got some things which we call inhibitors, which can bind to these ends. So an inhibitor is simply something that will be preventing the substrate from attaching to the enzyme. So inhibitors that attach to the active site, we call them competitive inhibitors. And then those that are attached to the allosteric site, we call them non-competitive inhibitors. And what competitive inhibitors will do is like in a vehicle, right? When you have got a vehicle and then someone sits on your seat, you can't sit there. So if a competitive inhibitor sits in the active site, a substrate can't sit there. But for a non-competitive inhibitor, when it attaches to the allosteric site, what it does is that it will change the conformation of the active site. So let's say the enzyme now becomes like this. Okay, the active site has changed because the non-competitive inhibitors attached to the allosteric site. So that's a problem. So when the enzyme attaches to the substrate, we form what we call the enzyme substrate complex. Now, this enzyme substrate complex will further break down and you still release the enzyme and also products. Okay, so the thing is that the enzyme is not changed in the reaction, but the substrate is going to be changed. You add this one piece, you can end up having two structures. Those are the products. It's not the same thing as what you had at the beginning. Now, what enzymes really do is that when you are talking about any reaction for it to happen, a reaction only happens when you overcome the activation energy barrier. Okay, from this point to the maximum point, we call that as the activation energy barrier. Every reaction, because here you have got reactants and then you have got products at the end. Before you can move from reactant to product, you need to overcome this mountain. And what an enzyme is really going to come and do is to help you lower this activation energy barrier so that you can easily form product, because once you've gone on top of the mountain you can easily slide it down. So that's why you need to lower that mountain to for you to climb. So what uh core enzymes do is that they are simply going to come and help the enzyme. All right, they are going to come and help the enzyme to fulfill its function. That's a function of a coenzyme. Now coenzymes, we have got what we call coenzymes. Coenzymes are actually biological molecules. And then we have got other molecules that are metallic in nature, for example, ions. Those are also going to be able to help in activating enzymes. But we'll discuss them in case I met any question on that. So question 1 is saying, what role does a coenzyme play in an enzyme catalyzed reaction? As we have said, it will be able to bind to the enzyme to enhance its activity. Okay. Number 2 is saying during competitive inhibition, the competitive inhibitor binds to either the active site or allosteric site. That's not true. Competitive inhibition is just binding to the active site. 2 is saying substrate binds to the allosteric site while the competitive inhibitor binds to the active site. This is also not true. The competitive inhibitor will actually bind to the active site, okay, but the substrate will not be able to bind to anything, because substrates bind to active sites. When the competitive inhibitor binds to the active site, everything is disturbed. There's nowhere to bind. 3 is saying competitive inhibitor binds to the active site. Okay, during competitive inhibition, this is true, isn't it? Competitive inhibitor binds to the active site. Active site is the only available. Active site is only available for the competitive inhibitor. This is not true because the active site is also available for the substrate. 5 is saying competitive inhibitor binds to the allosteric site, and that is false because that's for non-competitive inhibitor. Is this okay? Do we get the sense? Please, if you get it, just show me some reactions. Okay, with any emoji on your screen. All right, thank you. Number 3 is saying the active site of an enzyme consists of the catalytic site and the allosteric site, binding site and substrate complex, catalytic site and the substrate complex, binding and the catalytic site, binding site and the substrate complex. So the active site of an enzyme is made up of what? You can see to the active site, that is why you have got a binding site. Okay, that's why you actually find the substrate. So catalytic site and the allosteric site, there's no allosteric site, binding and substrate complex. Okay, of course the substrate is going to bind, yes, to the active site. Catalytic and the substrate complex, and then binding. Okay, so basically to the active site, that's where we're going to find the catalytic site, the site where we're going to, such that when the substrate binds it will be catalyzed, yes. So the catalytic site, yes, and also the binding site. Okay, so it's the binding and catalytic site. Number 4 is saying at some point in an enzyme catalyzed reaction, the rate reaches a maximum despite increasing the substrate concentration. Which hypothesis best explains the observation? So let's look at this before we even answer. Okay, the rate reaches maximum despite increasing the concentration. When you have got, say this is an enzyme, this is an enzyme, this is an enzyme, and then you have got substrates, they say they are like this. So at this point, because you have got the 3 enzymes, when you have only one substrate, the reaction is going to be slow. When you increase the substrates, the reaction is going to be faster. But when you exceed the number of substrates, when you exceed 3, at that point, even if you increase the substrates, the substrates that you are bringing in have got no enzyme to bind to. So at that point, the reaction is not going to increase. So on the factors that affect the rate of reaction, you have got the concentration of the substrate, the concentration of the enzyme, the pH, the temperature. Again, at the same time, when you have got, let's say, 50 substrates with one enzyme, the reaction is slow. When you start increasing the enzymes, when you reach 50, the reaction is going to be maximum. Should you go beyond 50 enzymes, the reaction will not increase anymore. Why? At that point, you increasing the concentration of the enzymes will not have the effect on the reaction because the enzymes don't have any substrate on which they should work. Okay, and you need to know for enzyme and substrate as a factor, what are you looking at? So for enzyme as the factor that determines the rate of reaction, you are going to have enzyme here and then the rate of the reaction here. Actually, let's swap them. You are going to have enzyme here and then the rate of the reaction here. What will be happening is that as you increase the enzymes, the rate of the reaction will increase. But there's going to be a certain point where even if you increase the enzymes, the rate of the reaction still remains the same, because at this point, what has just happened is the enzymes are increasing, but you don't have any more substrates. So when you're talking about now the substrate as the factor that will determine the rate of reaction, you are going to have substrate here and then the rate here. When you're increasing the substrate, rate is increasing, but there'll be a point when even when you increase the substrates, the rate of the reaction is no longer increasing. Why? Because the substrates are a lot, but you don't have enough enzymes. For temperature, the graph of for temperature as a factor and pH is a bit different. Okay, you are going to still have rate this side. Here we can have pH or temperature is the same. How these graphs go is like this. So here you can see when you increase the temperature of the pH, the rate of the reaction is increasing. So in this region, the enzyme is actually inactive. By the time you are reaching the maximum point, we call that as the maximum pH or the maximum temperature, or optimum pH and optimum temperature. That is the pH or temperature at which the enzyme works very well. At that point, you are going to have the maximum rate. But when you exceed the optimum, what will happen is that you start destroying enzymes. And so at that point, there is inactivation. That's why the rate of the reaction is slowing. Okay? Actually, not inactivation, but denaturation. You are destroying the enzymes. So at the beginning, the enzymes are not active. That's why the reaction is moving slow. But this side, the enzymes have been destroyed by too much pH or too much temperature. That's why the reaction is slow. Please know the difference. And then at this point, that's why I've got optimum or the best. Okay, so now we can answer question 4. At some point in an enzyme catalyzed reaction, the rate reaches a maximum despite increasing substrate concentration. Which hypothesis best explains this observation? The enzyme's activation energy has reached the threshold maximum value. The enzyme's activation sites are saturated. Now remember, here we are talking about what? Even when we are increasing substrate. So we're talking about this one. We are talking about the substrate. So for the substrate, remember, when you're increasing, it means that the enzymes have been all used up. Okay? So the enzyme's active sites are saturated. So this is the answer. Okay? I hope you understand that. If it was about the enzyme, the answer would have been the substrates have been used up. Does that make sense? Are we getting there? Okay. Number 5 is saying, which of the following statements about DNA is correct? It's made up of a single twisted polynucleotide strand. When you look at the structure of DNA and RNA, RNA is like this. It is a single stranded molecule. Now DNA is like this. It is helical. It is a double stranded molecule. And the double strand is because of hydrogen bonds. Adenine is attached to thymine by two bonds, while cytosine is attached to guanine by triple bond. Okay. In RNA, you don't have these bonds. Actually, also in RNA, you don't have thymine, but you have got uracil there. So, which statement is correct? Is made up of a single twisted polynucleotide strand? That is not true. Nitrogenous bases and phosphates form the backbone of the molecule, while the sugars point inwards. So, when you look at this one, the way DNA is really is like this. This is your nitrogenous base. This is your pentose sugar. You have got a phosphate. Again, you are going to have a phosphate here with a nitrogenous base. again, and this will continue. Another molecule is going to be this side, but in this side it will be moving in the opposite direction. Hope you can see the way I'm drawing it. It's moving in opposite direction. So the end where a phosphate is, we call that as the 5' end. And then this point here, we call it the 3' end. For the other strand, the 5' end is the one down, the 3' end is the one there. So the structures that you can see this side, we call it a backbone. The backbone is this part and this part. The internal structure is actually this part where I've got hydrogen bonds. Okay, so the option 2 is saying nitrogenous bases and phosphate form the backbone of the nucleotide while the sugars point inwards. Now you can see the sugars are not the ones pointing inwards. It's actually the nitrogenous bases that are inside. These are the nitrogenous bases. Okay, the sugar is this one, the phosphate is this one. These are the ones forming the backbone. 3 is saying it is made up of two strands whose polarities face in the same direction. And that one is not true because they face in opposite directions. Number 4 is saying in animal cell it is highly compressed into tight structures with aid of histons. And that's correct. Okay. For example, in us. Number 5 is saying it contains combination of 4 different bases with an equal number of purines and pyrimidines. This statement is false because you actually have equal number of purines and pyrimidines. For example, if you have got adenine, you're supposed to have thymine. Adenine, remember the purines. Purines, we can say pure as gold. PAG, purine as gold. So purines have got adenine and guanine. For pyrimidines, for me I use put C, put C. Pyrimidine, you have got uracil, thymine, and cytosine. So it's just that in DNA you don't have uracil. So you only have these two, thymine, cytosine. So when you have got one purine, you're supposed to have one pyrimidine. If you have got two, you're supposed to have again two pyrimidines. They should be equal. Someone has written a question in the chat box. Please feel free to ask in case you have got a question. Okay? Anyone who has got a question, have we understood? Anyone with a question? So question 4, the answer is option 2. Enzymes active site are saturated and additional substrate cannot increase the rate of the reaction. Number 6 is saying choose a combination that represents a nucleoside. Now remember, when you have got this guy, which is a pentose sugar, attached to a nitrogenous base, we call this as a nucleoside. This bond between a pentose sugar and a nitrogenous base, we call it the N-glycosidic bond. Okay, this is a nucleoside. Now to this nucleoside, if we add the phosphate plus phosphate, it becomes a nucleotide. Okay, we now add the phosphate. So we want to choose a combination that represents a nucleoside. Cytosine and guanine, these are both bases. Guanine and phosphate, no. Ribose and phosphate, that's also a no. Deoxyribose and pentose. Deoxyribose is a pentose sugar. Pentose is also a sugar. Deoxyribose and cytosine. This is correct because deoxyribose is a sugar, cytosine is a base. So a sugar and a base is what makes up a nucleoside. I hope that makes sense. Have I left out anyone? Are we okay? Please, again, I'll say feel free to ask if you're not clear. 7 is saying, which component of DNA determines the genetic code? Now, what determines the genetic code are the bases. Nucleotide, these ones, the nitrogenous bases. Okay? So it's not a sugar phosphate backbone. The backbone, remember, we said is this. It's not the backbone. The sequence of all nitrogenous bases ATCG. Now, on the genetic code, we don't have thymine because the genetic code is actually on mRNA, messenger RNA. So we don't have thymine. The sequence of nitrogenous bases AUCG. That's correct. Okay. A code of two bases. Now, the genetic code is not two bases, it's actually three bases. In case you don't understand much about the genetic code, just search on YouTube the genetic code, Acidius Academy. I've explained it so much in detail and answered a lot of exam questions at the end, right? Number 8 is saying, which of the following statements is incorrect about the structure of DNA? DNA chains are typically found in a double helix form. That is correct. In a double helix form, two complementary chains are bonded together. This is also true. They are bonded by hydrogen bonds, the complementary strands. Complementary means that if you have got adenine, it's going to pair with thymine. If you have got cytosine, it will pair with guanine. That's the meaning of complementary. The sugars and phosphates lie on the outside of the helix forming the backbone. This is correct also, as we said. Number 4 is saying the nitrogenous bases extend into the interior like the steps of a staircase in pairs. And this is correct, as we also mentioned. Nitrogenous bases of a pair are joined to each other by covalent bonds. This is false. They are joined by hydrogen bonds. And please don't forget that A and T, two hydrogen bonds. C and G, triple bond, which is hydrogen bonds. That is why during replication or transcription, we separate the strand where you have got a lot of adenine and thymine because it is easier to separate double bonds than triple bonds. Number 9 is saying, question 7. Components of DNA which determine the genetic code. So the genetic code is read on mRNA. For example, when you see 5 prime A U G C C, maybe U A C C G 3 prime. This is what we call the genetic code. It's found on mRNA. What determines the genetic code is basically the bases which are found in RNA, except thymine, because thymine is not found in RNA. Does that make sense? Uh, yes, please. On on question what? Yes. What's the question? Okay. I get you. I get you. So the question is actually saying the component of DNA determines the genetic code. So I also quickly rushed through and didn't even see that DNA. So because it's DNA, you're supposed to have ATCG. Okay, but the genetic code itself doesn't contain the thymine, so you're supposed to have uracil. So because it's DNA, please make that correction. You're supposed to have the thymine there. Thank you so much for the observation. Excellent. Okay. So number 9 is saying the dash modifies the package protein and the dash modifies and packages protein and lipid molecules. Mitochondria, lysosomes, Golgi apparatus, smooth endoplasmic reticulum nucleus. Now the mitochondria is the powerhouse. It's involved in what we call aerobic respiration. Okay, so this one is for ATP production. It is involved in respiration. And then a lysosome, these ones contain what we call hydrolytic enzymes. Hydrolytic enzymes break down things. They break down different things that are going to be taken by cells. For example, if a bacteria enters your body, it is going to be engulfed, it is going to be eaten by what we call phagocytic cells like neutrophils. Okay, and then once it is been eaten, lysosomes will produce these enzymes to be able to destroy that bacteria. The Golgi apparatus is involved, this one is involved basically in processing. Okay, and what it is really processing is when you make proteins, those proteins, because what happens that the rough endoplasmic reticulum will make proteins. Those proteins are not really active, so they have to go to the Golgi to be activated. The smooth endoplasmic reticulum is involved in the formation of lipids. Okay, hope that is correct. Hope that is clear. So the dash modifies and packages proteins and lipids. So smooth endoplasmic reticulum, of course, it will be able to modify. It's actually involved in the formation of lipids, but modification is the Golgi. I hope it's clear. Making lipids smooth ER, making proteins rough ER, but modification is the Golgi and packaging them so that they can be transported to where they are needed in the body. Prokaryotic and eukaryotic cells are similar in that they both, what you need to know is that prokaryotes, y- yes, please. So the answer is 3. Golgi apparatus. Lysosomes produce hydrolytic enzymes which destroy microorganisms. Okay, does that make sense? Very good. So what you need to know about prokaryotes is that there is no membrane bound organelles. So prokaryotic cells don't contain any organelle that has got a membrane. Examples of such organelles, nearly all. Okay, the only, please know this. Most of the organelles that you can ever think of, mitochondria, lysosomes, Golgi, smooth endoplasmic reticulum, rough endoplasmic reticulum, nucleus, all those have got a membrane, meaning that a prokaryotic cell can't have them. Eukaryotic cells have got membrane bound organelle. The only organelle that does not contain a membrane is a ribosome. That is why a prokaryotic cell contains a ribosome, because it doesn't have a membrane. A ribosome is also going to be found in eukaryotic cells. So because prokaryotic cells do not have membrane bound organelles, it means that they also don't have a nucleus because it also contains a membrane. So now where is DNA in a prokaryotic cell? DNA is just found freely in the cytoplasm, and we call it the nucleoid. Nucleoid. Okay? While eukaryotic cells will contain the nucleus, and the middle of the nucleus that is dark, we call it the nucleolus, which contains the genetic information. I hope that's clear. So prokaryotic and eukaryotic cells are similar in that they both, so the similarities is that they both contain cell walls, cell membranes, ribosomes, cytoplasm. So contain mitochondria, false. Contain ribosomes, true. Have a nucleoid. Nucleoid is only present in prokaryotic. Example of prokaryotic is a bacteria. An example of a bacteria is E. coli. A E. coli is an important bacteria that affects females. Females, most of, especially you guys in university where you are using, these toilets, a lot of people, you are prone to having UTIs. It's because of E. coli. And then three, have a nucleoid. In case you're wondering what is E. coli, maybe some of you know. No, I was told at the clinic I've got yeast infection. Okay, such. All right, we're not teaching medicine, we're teaching biology. So the other options are not correct because they're not found in both. Plasmid is only found in prokaryotics, is circular DNA. 11 is saying choose the false statement about the nucleus in animal cells. It is surrounded by cellulose wall. It's made up of nucleic acid and protein. It's surrounded by a membrane. This membrane is perforated. It's the largest organelle. So choose the false statement. Largest organelle is true. Its membrane is perforated is true. A nucleus contains what we call nuclear pores. So that when the DNA is converted to RNA, that RNA can move out to the nucleus to go into the cytoplasm to undergo translation. It is surrounded by a membrane, that is correct. It is made up of nucleic acid and proteins, that is correct. It is surrounded by cellulose wall. This is false. What surrounds the cellulose surrounds the cell membrane, not the nucleus. And it is actually the cell membrane of animals. Sorry, plants. Choose a combination that represents a nucleoside. This question has been repeated again. Okay, this page is actually just a repetition. Number 12, pili. Now, when you look at bacteria have got pili. Pili are basically these structures which help bacteria in movement. Another important function of pili is that it is involved in reproduction. Okay, for bacteria, they don't have any sexy cells, so these will be acting as the things of reproduction. Another important thing about pili is that, you know, this is a bit of medicine, but let me tell you, some bacteria are resistant to drugs. That's why when you go to the hospital, they'll barely give you what we call antibiotics, drugs against bacteria, because most of the bacteria are resistant. And the more you take antibiotics, the more you become resistant to these drugs to the point that when you really need the drug, it will not be able to work. You'll die. These things are very bad. So what happens that when one bacteria is resistant to some drugs, it can transfer those genes that code for proteins to make the bacteria to be resistant to another bacteria via the pili. Hope you get that. So pili in bacteria can be best described as the outer covering, thread-like projections for movement, thread-like projection on the surface for attachment, vesicle in the cytoplasm, organ involved in cell division. So these are thread-like projections, okay, for movement. Now for movement, it's different. You have got one which is very long, we call it flagella. The flagella is the one involved in movement. The others, the small ones, the pili, those are involved in attachment. Okay, we call them as virulence factors. What's the meaning of virulence? Virulence is the ability to cause disease. I hope that's clear. Number 13 is saying, which of the following enzymes synthesize RNA primers? Now, kindly just switch off your mic if it is on. When you have got DNA, remember DNA is like this. During replication, you want to make a copy of this guy. The first thing is that the way this guy is like this, it will be very hard for you to separate it. You need to make it like this. So we have removed the supercoils. The enzyme that removes the supercoils, we call it DNA topoisomerase. The other name for topoisomerase is DNA gyrase. Gyrase to remove supercoils. And then what will happen is that another enzyme will come in, it will start separating this strand. And so what you'll be able to see is this. And this enzyme that is separating the two strands, we call it helicase, which is this one. Now what helicase is going to be doing is that it is separating the strand. The direction that helicase is going, we call it the direction of the replication fork. Replication fork. Okay. But then the thing is that this side where you are separating the strand, these ends, these guys can still come back together. So to prevent a coming back together after separation, there's another enzyme which is known as SSB, single-stranded binding protein. This one will basically stop the coming back together of the separated strand. As this is happening, another enzyme which we call DNA polymerase 3 is going to be coming in. But before DNA polymerase 3 comes in, you have got what we call primase. Now you have got two strands here. The strand that is reading from 3' to 5', and the one that is running from 5' to 3'. The one from 3' to 5', we call it the leading strand. The one from 5' to 3', we call it the lagging strand. So on the leading strand, RNA primase is going to come and lay down one primer. A primer is just an RNA nucleotide. While on the lagging strand, RNA primase is going to lay down a lot of primers. A lot of primers. Okay? What is going to happen on the leading strand is once you lay down one primer by the RNA primase, DNA polymerase is now going to come and continue forming phosphodiester bonds, forming a new strand. The strand that is being formed is running from 5' to 3' because the strand that we are reading is from 3' to 5'. Remember, DNA polymerase can only read the strand from 3' to 5'. So meaning that for the lagging strand which is running from 5' to 3', DNA polymerase can't read it. So in that case, DNA polymerase is going to be working in the opposite direction. That is why DNA polymerase is bidirectional. But on the lagging strand, it will not be forming phosphodiester bonds. Instead, it will just be forming short bonds, connections, and we call them Okazaki fragments. Okazaki fragments. What will be now coming to connect these short fragments, which we call Okazaki fragments, on the lagging strand, is what we call DNA ligase. DNA ligase. So I've basically described for you the entire process of DNA replication. I hope it's clear. So the question is, which of the following enzyme synthesizes RNA primase? So primase is by primase. Okay, remember primase are RNA molecules. Does that make sense, guys? Very good. Number 14 is saying, during elongation of DNA replication, the enzyme dash catalyzes the joining of deoxyribonucleic ribonucleoside 5' triphosphate to the growing DNA chain. Now, I've already mentioned this. DNA polymerase is the one that to catalyze the formation of these DNA strands. Okay, it's DNA polymerase 3. So what DNA polymerase will just be doing is once you lay down a primer, DNA polymerase is going to come and continue making that primer long so that you form a long chain of DNA. 15 is saying a defect in the enzyme DNA ligase during replication would most likely result in which of the following: impaired separation of parental DNA strand, continuous synthesis of the leading strand but discontinuous lagging strand synthesis, failure to remove RNA primers from the newly synthesized DNA strand, an inability to connect Okazaki fragments on the lagging strand, and an increase in the formation of a single-stranded DNA region. Remember, the function of the DNA ligase is to come and connect the Okazaki fragments via phosphodiester bonds. Therefore, if you don't have DNA ligase, you expect that there's going to be an inability to connect Okazaki fragments. Remember, Okazaki fragments are formed by DNA polymerase, but connecting them to form the phosphodiester bonds is by ligase. In DNA replication, the lagging strand is synthesized in the 3' to 5'. This is true, isn't it? Remember, as I said, the leading strand moves from 3' to 5'. Therefore, the strand that is going to be made from the leading strand is going to be 5' to 3'. At the same time, the lagging strand is from 5' to 3'. Therefore, the new chain that is going to be synthesized from the lagging strand is going to be from the 3' to 5'. Okay, that's a thing. So in DNA replication, the lagging strand is synthesized in the 3' to 5' direction, synthesized in the direction of the replication fork. So you don't make it in the direction of the replication fork. Look at the way I've written an arrow. It is going downwards. It is away from the direction of the replication fork. Synthesize in the direction of the replication fork. No. Okay. And then 3 saying is synthesized in the direction away from the replication fork. This is true, isn't it? Does not require primers. It requires actually a lot of primers. Synthesized continuously. No, it is discontinuous. So is synthesized in the direction away from the replication fork is correct. Number 1 is saying is synthesized in the 3' to 5' direction. Would this exclude this because probably they are referring to the strand itself that you are reading. Otherwise, the strand that you are making, of course, is going to be made from the 5. You are reading the chain from 5' to 3', meaning that the new strand you are going to make will be from 3' to 5'. But for this question, there's actually a better option, which is option 3. Just to avoid maybe they were trying to refer to the strand that you are trying to read. I hope that is okay. Okay. So there is no debate on option 3, but we cannot debate on option 1. That's why it's not a best option. But if option 3 wasn't there, I would pick option 1. Number 17 is saying which of the following is not a characteristic of the genetic code? Yes, please.
Yeah, according to the question you have explained, so how possible like when you pick one, they could not even.
If you pick one, it's wrong. Yes, in this case. If you pick it, it's not correct. All right. So which of the following is not a characteristic of the genetic code? Okay, specificity is not part. Universal. The genetic code is the same for organisms. Again, please just search on YouTube genetic code Acidius Academy. You understand everything you need to know about the genetic code. It is universal because it's the same for organisms. It has got triplet codons. It's degenerate. The other term for degenerate is it is a redundant. And then number five, it is nano. Okay, this is overlapping. I'll now change. It is non-overlapping. Okay. The thing for specific, this when you read all the properties of genetic code, specificity is not really included. But because there is overlapping is really wrong. The genetic code does not overlap. It's non-punctuating, non-overlapping, commaless, degenerate, unambiguous, redundant, triplet codon, universal. Those are the properties of genetic code. Okay. 18 is saying which of the following molecules contain introns. Now, when you are making in prokaryotic cells, right? When you synthesize the mRNA that you synthesize is actually going to have, let's make it like this. So I'm trying to make introns and exons. So the ones in blue, we'll call them exons. So what happens is that once transcription ends, you have got what you call because transcription is the formation of RNA. What you form is known as pre-mRNA. It is immature. Okay, it is immature. So what happens in that case is that three main modifications have to happen. Number one, there's what we call splicing of exons. So to splice is to join. What is happening is that we are going to remove the introns and then join the exons because introns are not required. Number two, there's what we call 5' guanosine triphosphate cap. This is a cap and it is being added on the head. So the guanosine triphosphate is actually added on the head. Number three, I've got what we call 3' polyetyl. In other words, the mRNA that you formed to the 3' end, which is the tail, you are going to add a lot of adenines. So these are the modifications that happen. All right. So which of the following molecules contain introns? Primary mRNA transcripts of prokaryotics, primary mRNA transcript of eukaryotic cells, primary tRNA transcript of eukaryotic. Now, first you need to know it's mRNA, and it is the mRNA of a eukaryotic cell, which will undergo these modifications. Okay. Number 19 is saying RNA processing takes place during translation, occurs during DNA replication, takes place before transcription. Now, RNA processing is what I've just mentioned here. It will happen when you've made RNA. So this happens when RNA is being transcribed. Is an event that occurs after RNA has been transcribed or when RNA has already been made. More than one codon may specify the same amino acid. This feature of the genetic code is called degenerate. The other term for degenerate is redundancy. The genetic code is redundant. The meaning is that you can have one amino acid, but you have got a lot of codons. Okay, maybe you have got let me not use, anyway, I can even use this one. These are stop codons, UAA, UGA, UAG. You can see these are different codons, but they are specifying the same thing. It's just that stop codons don't specify any amino acid. These three I wrote are stop codons. The start codon is UAG. UAG no. UAG, UGA, UAA. These are stop codons. You can have, let's say for example, CCC, CCU, CCG. All these guys will give you the same amino acid. That's the meaning of degenerate. So the third position can be different. And because the third position can be different, but you are still coding for the same amino acid, we call that as the third position WUBOs. So we call that as the WUBO hypothesis. The third position can be different, but you are forming the same thing. Another thing you need to know is what we call non-ambiguous. Non-ambiguous means that, look, you can have one amino acid being formed by a lot of codons, but one codon can't give you two amino acids, or it can't give you more than one amino acid. One codon can only give you one amino acid. That's the meaning of non-ambiguous. Okay? I hope it's clear. 21 is saying which of the following is an incorrect statement about the genetic code. It has codons in which the nucleotide of mRNA are arranged in a linear sequence of successive triplets. That's correct. It is always read in 5' to 3'. Very correct. It is the same for most organisms. Very correct. That's the meaning of universal. Some amino acids are specified by more than one codon. That's correct. That's the meaning of degenerate. Some codons specify more than one amino acid. This is false. This is against the non-ambiguous. These statements would mean that the genetic code is ambiguous, but it is non-ambiguous. So a codon can't specify more than one amino acid. 22 is saying during the process of translation, each target amino acid is activated by. So what happens is this: you have got your this bro which we call tRNA. Let me just try to draw it professionally. The structure is something like this. The 3' end, this is where the amino acid is going to attach. But for the amino acid to attach to the tRNA, you need an enzyme which we call amino acid tRNA synthetase. And that process, the attachment of an amino acid to tRNA, we call it tRNA charging. But the amino acid, before it is attached to tRNA, you need to attach energy to it, ATP. So we call that amino acid activation. And all that is carried out by the enzyme which we call amino acid tRNA synthetase. Okay, so during the process of translation, each target amino acid is activated by ATP using the enzyme amino acid tRNA synthetase. Please notice synthase, synthetase. If you hear synthetase, it means that energy is involved. Synthase means that there is no ATP involved, which is energy. Okay, I hope that is clear enough. So let me read again. Each target amino acid is activated by ATP using the enzyme. Okay, tRNA using the. All right, that's correct. The option 5 is saying tRNA using the enzyme amino acid tRNA synthetase. The attachment of amino acid to tRNA, that is not really activation. We call that tRNA charging. 23 is saying the promoter is a binding site for. Okay, you have got what we call the promoter. This is the lac operon. Lac operon is just a structure on your chromosome that regulates the breakdown of lactose. So the promoter is where what we call the repressor. So we have got lac repressor, RNA polymerase, DNA polymerase. We have got what we call the promoter. We also have what we call the operator. So we need to know basically what this guy, the role of these guys. So again, Firstly, first thing first, what is the lac operon? The lac operon is just a piece of the chromosome that is going to be involved in regulating the breakdown of lactose. What happens is this: in our bodies, the primary source of energy that we use is actually carbohydrate. Sorry, no, glucose. But in a state where all the glucose has been used up, where are you going to get energy in that case? Well, you don't know. There's nowhere where you're going to get energy. So I want to show you something. I hope you'll be able to see it. Okay. Yeah. Okay. This is not what I want to show you. This is gene regulation. Okay. I can't even put that one here. So what I'm trying to say is, when you use up all the glucose, okay, you've used up all the glucose in the body. In that state, you don't have any other source of energy. So if you drink milk, milk contains lactose. So in that case, you can get some lactose from the milk. And that lactose now can be broken down into the glucose. And that glucose is now what you can use for the energy that you need. But you need some enzymes to help you in breaking down that glucose. Okay? Where are those enzymes going to come from? That is where the lac operon comes in, to help in the production of those enzymes that will help in this process. So we have got what we call the promoter, we have what we call the operator, and then at the end here, we also have what we call the repressor. Now, the repressor is actually an enzyme that is going to be produced. So, remember when you're talking about that is transcription, we have got a promoter. The promoter is where an enzyme DNA polymerase is going to come and attach. Okay. But what happens is that before this can happen, you are going to have a gate. Let, let me put this as the operator, and then the promoter is in front. I want you to understand this, Kamaga, is an interesting one. So polymerase, DNA polymerase comes to attach here. Okay. So not DNA polymerase, but RNA polymerase, because here we form RNA in this case, so that we can form proteins at the end. Now the operator will bind what we call the repressor. The function of the repressor is to stop, because the RNA polymerase is actually here. It is moving along this structure. So as long as the this guy repressor is attached to the operator, RNA polymerase can't pass. Once the repressor moves out of the operator, it will mean that now the operator is free. This structure is no longer there because it's acting as a switch. It has switched off this structure, which we call the lac operon. So once the repressor is out of the operator, the way is now open. RNA polymerase can go and attach to the promoter, and then we'll be able to form the enzyme that will be used to break down your lactose so that you can have glucose at the end. So that was just me trying to attempt to explain for you the lac operon. But at the end here, you have got three structures. We have what we call the lac A, the lac Z, the lac Y, which will produce three different types of enzymes. Again, you can just search lac operon Acidius Academy. You'll be able to understand it very well. So we've said the promoter is the site for the attachment of RNA. polymerase. 24. Which of the following is incorrect about lac operon in E. coli bacteria? Required for the transport and metabolism of glucose, not glucose really. Lactose is the inducer molecule. Glucose is co-repressor. Now the thing is, I'll mention again, when you have got glucose, it means that the lac operon is off. Okay? Glucose is a repressor. It's a co-repressor. Now lactose, the absence of glucose and the presence of lactose is what will switch on the lac operon. So lactose is actually an inducer. When the operator gene is switched off, no mRNA is transcribed. That is true. When the operator is off, and it is off when the repressor is attaching on it, because at that point the RNA polymerase can't pass. When lactose is not available, the lac operon cannot be expressed. That is also correct. Okay, so what is incorrect? Lactose, glucose is a co-repressor. That's true. One is saying it is required for the transport and metabolism of glucose. It's not metabolism of glucose but lactose. That's why this one is all not correct. 25 is saying which of the following is false statement of the lac operon? Lac operon is activated only when glucose is absent. That is false. It's not only. It's when glucose is absent and then lactose is present. That's when it gets activated. Okay. So which of the following statement is false? So one is false. Two is saying lac operon is switched off when glucose is present. Yes. Lac operon is activated when glucose is absent and lactose is present. Yes. Lac operon is activated when glucose is present and lactose is absent. So activated when glucose is present and lactose is absent. Okay, what is the difference between these two? Lac operon is activated. So it is going to be activated when glucose is absent, yes, and lactose is present. And then for lac operon is activated when glucose is present and lactose is absent. This is false, so I'll take it. Lac operon is activated when both glucose and lactose are absent. Again, that is not true. It is going to be activated. Of course, it's only activated when lactose is present but glucose is absent. Okay. So which of the following is the false statement? So when you want what is false, well, there are many false options here. But four, I think, is the most important. Lacoperon is activated when a glucose is present. As long as glucose is present, lacoperon is inactive. Okay, so that's the best that we can pick for here because it's just a direct opposite of the required statements. Of course, the other options also have got something wrong in them, but option 4 is the exact wrong thing. 26. In the regulation of the lacoperon, what would be the expected outcome if the operator sequence were deleted? If the operator sequence were deleted, operon would be permanently repressed. Remember I said the operator is what prevents the polymerase, RNA polymerase from moving. So if the operator is not there, it means that there will be continuous production of these enzymes. Okay, the operon would be permanently repressed, it wouldn't be replaced. Structural genes would be continuously transcribed. That's true. The structural genes are the genes that are going to be coded by the enzyme I told you that, by the genes I told you that you have got. So for the lac operon, this is structure like this. You have got two genes. You have got what we call like lac inducible, and then you have got the operator and the promoter. These ones, these are controllers. And then we have got structural genes where I've got like A, like Y, and like Z. These are the ones that will be producing the enzymes. So these ones we call them structural genes. Okay. The repressor protein would bind more tightly to the promoter. In that case, the repressor wouldn't be able to, because at this point, remember, the operator is not there. That's where the repressor binds. The operon would only function in the absence of glucose and lactose, and that wouldn't also be true. RNA polymerase would fail to bind to the promoter. In this case, it would always be binding because the operator is always open to allow the RNA polymerase to bind to the promoter. Okay. 27. Dash substitution is a type of gene mutation where a purine is replaced by a pyrimidine, and this as the dash on the protein synthesized. You have got transverse mutation, transitional. Okay. Are you still able to get me? Just confirm with me. Okay, just a second. I just try to check my connection. Sorry, it just seems my connection is just gone down. But since we are nearly done, we'll be able to pause here. Okay? The class will be able to find it on the drive for the two questions that are remaining. So let's be able to answer the questions now that are remaining. So the question is saying, dash substitution is a type of gene mutation where the purine is replaced by pyrimidine, and this has a dash on the protein synthesized. Okay, is replaced by pyrimidine. What is really happening in that case? What type of mutation is that one? Now, for the mutations where you have got a purine, so we have got what we call transverse, okay, and also, sorry, we have got transverse and transitional mutation. So we need to know the difference between these two. So this is just when you're talking about let's say you have got what example of purine? Okay, we can say purine, and then pyrimidine, and then pyrimidine. Okay. So if you change a purine for a purine or a pyrimidine for a uh, pyrimidine, this is transitional. But purine for pyrimidine, pyrimidine for purine, or the other way around, that is transverse. Okay, transverse. And for that, for the transverse, there's going to be significant negative effect. Okay, because you've really changed everything. A purine for a pyrimidine is totally making everything different. 28 is saying which of the following point mutation causes frame shift. Now, a frame shift mutation is a mutation where there is a change in the reading. Let's say, for example, you had A, C, U, C, C, G. So when you are reading this, you're supposed to read this first three and then you read this first three. On the genetic code, this might give you amino acid A, this might give you amino acid B. But let's say you put another nucleotide here. So now you have A, C, let's say another C is added. And then you have got U, C, C, G. So in this case, everything will change. You will read ACC. This will give you a different amino acid C. Then you read UCC. This will also give you a different amino acid B. I mean, let's say D. Okay, this is what we call a frame shift. There is a change in the way you've read your chains. That's a frame shift mutation. But if we added three nucleotides on the middle here, that's not a three. So addition of three nucleotides or deletion of three nucleotides is a non-frame shift mutation, because we're simply adding a different amino acid on the mid or between, or removing an amino acid, and not really changing the way you read. So which of the following point mutations causes a frame shift? You have got transverse substitution, insertion of one base. Yes, the insertion of one base is going to cause this one. Okay, deletion of three. When you delete three or add three, it's not frame shift. Nonsense mutation is where you basically change. It's a point mutation. Let's say if you add A, you end up changing. You add, let's say, CCA, you change and then you have got CCG. But this one and this one, they are still giving the same amino acid. We call that as a nonsense mutation. So in this case, the answer is this one. 29. A genetic mutation in which a stop codon is prematurely introduced into the coding sequence of a gene would result in. When you have got a stop codon being introduced, we call that as a, of course, it's nonsense. It's a nonsense mutation. That's what is known. So this one is a nonsense mutation causing a premature termination of the protein chain. 30 saying the mutation that causes sickle cell anemia in human is caused by. Now, for sickle cell anemia, I've got at position number 6. Okay, position 6 on the beta gene of hemoglobin. What will happen is that you substitute valine for glutamate. Okay, so that is what happens. So what type of mutation? A base deletion, a base addition. It's a missense mutation. Why do we call it missense? Because you substitute one base for another, and then what you end up producing is a totally different amino acid. So where there's supposed to be valine, now you have got glutamate. Okay. If it was giving us the same amino acid, we call that a nonsense mutation.